Thank you, David, for the kind words, and also thank you for the organizing committee to invite me to speak about research that my group's been involved in for a number of years, and I'll try to really synthesize some results on studying proteins in the realm of diagnosis and theranostics. This slide really shows a main motivation. Why are we interested in studying proteins? And many pathologists in the audience, I don't need to explain to you, we all understand that proteins are really the machine or the engines of life as well as disease. They carry out functional chemical transformations, biochemical transformation, metabolic transformations, and so forth. And really all, if you think about it, all new targeted therapies that are being developed are in one way or another directly binding to proteins and perturbing protein signaling. And so why not study proteins directly? We also know that if we understood aberrations at nucleic acid level in a very detailed manner, we may still not understand how phenotypic changes occur and how different diseases respond to therapies. And that's illustrated here on this slide, where if you look at identical genomes, in this case identical twins, they have different phenotypes. They have different fingerprints, for example. And so that's by analogy, of course, disease that may look at nucleic acid level similar or very similar may not behave clinically and therapeutically the same way. And so that's yet another reason why we're very interested in studying the machines of life, if you will, directly. If you think a little bit about what experimental technologies are available to study proteins in model systems as well as in humans, you can sort of naturally think about it more in liquids, sort of circulating fluids, urine, plasma, blood, CSF, and others, and in tissue. And so there are, of course, assays to assay functional capability of enzymes and proteins directly, and we do that in laboratory medicine very successfully with enzymatic assays, directly testing the chemical capability of a protein, like transferases and so forth, aminotransferases, for example. We also do this in tissues. It's been done for many, many decades, looking at cholinesterase, for example. There are also ways how to look at proteins in tissue, not through antibody-antigen interactions, but directly through chemical modification. For example, the Roehrl stain is a good example where a chemical reaction takes place. And then there's a whole category that you are very familiar with, and that's using antibody-antigen interactions with all its advantages and disadvantages. And in laboratory medicine, of course, technologies like Phenomimetry, ELISA, immunoassay in solution, very successful. In tissues, immunohistochemistry, immunofluorescence assays, and other things that use other readouts like ion beam imaging rely on an antibody-antigen interaction and then use variable readouts to quantify the degree of interaction and therefore then conclude the amount of pro-target analyte that may be present in the tissue that you're interested in. And then there's a whole group, and that's what I'll be focusing on in the talk primarily, of technologies that look at the abundance and chemical modifications and aberrations of proteins directly in the sample that you'd like to analyze without the need of an antibody-antigen interaction. And they rely often on liquid chromatography, mass spectrometry, and other types of technologies. And again, technology is only as good as the question you're asking. I think this is another very important point I want to make. This is not just a talk about technology. Actually, the rest of my talk will be very much about applications and trying to understand disease and discovering biomarkers and validating them. Because technology evolves very rapidly. I think one of the advantages we have in pathology is that we can be at the cutting edge and we can evaluate what are the best technologies to ask and answer the medical and therapeutic and theranostic question we would like to answer. So again, the point really is we don't want to be stuck in one technology only. The question, the technology is really only the tool to answer the question. The question is key. So how do I think about a biomarker? People talk a lot about biomarkers. I think about it in the following terms. I think it's a defined characteristic, so it's a molecular entity, if you will, that we measure, and what I measure is very quantitatively with an error associated with knowing what the characteristics of the measurements are. So really this is not semi-quantitative, this is really measuring femtomoles per milligram of tissue, for example. It's an indicator of either a normal biological process, so there are many biomarkers that are very useful for that, of course. It can be something that describes a pathogenic process, and increasingly, I think, what I'm really also interested in is markers that can be used to study the time evolution of disease under perturbation, and perturbation can be a targeted therapy, it can be radiation, it can be a combination of interventions that we as physicians make. And trying to use biomarkers to study that process is something that's obviously very appealing, but quite hard to do. But I think it's really the future of where a lot of pathology is going in the next couple of years. So in my talk, I want to talk now really about applications that I have been involved in, that my lab's been doing over a number of years, and show you strategies of how to look at proteins in various human diseases using, again, different flavors, different flavors of proteomics to look at proteins. So we'll talk about enrichment strategies. We'll talk about post-translational modification, such as glycosylation. I'll show you a really important application of that, why that's important and why that's something you can't really get by just looking at nucleic acids. I'll show you an application of something that I've been very interested in recently, and that's using, if you will, autoantigen and autoantibody characterization in cancers. And that's really sort of an IO application, but comes out of autoimmune disease research we did some while ago. And then I'll briefly talk about what we call unbiased deep proteome profiling, which is sort of the analogy of whole genome, whole exome sequencing, but applied to looking at the protein domain in human disease. And finally, applying that to clinical trials. And I'll show you some initial work that we've been doing to try to really develop technology that can be plugged into an ongoing clinical trial and then help co-develop the technology as well as informing the trial, hopefully in terms of therapeutic options. So a huge challenge really in proteomics has been that A, there is no PCR equivalent in proteomics. You can't just go and amplify proteins at will. That's not possible. You can't, I mean, it's just fundamentally not possible. And the other problem is that proteins in human tissues and biofluids exist at very large dynamic range, about the order of 10 to the 10. Actually, IL-6 is about the most least abundant protein in plasma, albumin the most abundant, and they have a 10 to the 10 concentration difference physiologically. And any single technology that you think about really can't handle 10 orders of magnitude. So that's a problem. And so one approach to do that, and that's really what biochemists would do, is to simplify the problem and fractionate, and you're only looking at a subset. And you enrich for things that are rare, depleting things that are abundant. And I'll show you two examples, one using a heparin affinity enrichment strategy, and another one really looking at glycosylation of proteins using lectin pulled down in lectin arrays. So when we did that, and I can't go into too much technical detail here, but when you enrich subfractions of tissue proteins, you can then actually start to see a lot more diversity, you see a lot more proteins. So each of these spots here on such gel is a single protein. And we saw by enriching that we started to see less abundant proteins more robustly, and we started to see about 15 and sometimes higher fold enrichment and number of detectable proteins that we could see. So that's very helpful. And then you start comparing essentially protein expression patterns in normal colonic mucosa, for example, versus invasive adenocarcinoma as a simple example. And you can use mass spectrometry then to ask the question, what are the entities of the proteins that are expressed in normal colonic mucosa versus the entities, the molecular entities that are expressed in adenocarcinoma of the colon? A very simple question, but you really need sophisticated technology to make the assignments of proteins that you a priori have no exact knowledge about what they are. And the way this is done is by measuring peptides. Again, not too much technical details, but you're measuring peptides that you extract, that you modify, that you measure the mass of very accurately, that you can actually sequence. You can cut off one amino acid at a time, measure mass differences, and through this you can deduce the amino acid sequence of particular peptides that you then map back onto the amino acid sequence of the protein. And if you do that successfully, you can confidently identify an identity of a protein that you found. And you can do this over and over, literally with thousands of proteins. In addition, you get actually some relatively good quantitative information that is rather linear, and which distinguishes this technology from many others that aren't really good linear transfer functions between what you put in and what the readout is. So here we have a pretty good idea about, across about 5 to 6 orders of magnitude, what the amount of that protein present in the sample in the tissue is. So this is an example just to show why enrichment is useful. So in this particular study, we showed that about 60% of protein biomarkers that differentially characterized invasive adenocarcinoma of the colon from normal colonic mucosa were only visible after doing a good biochemical prefractionation approach. If you had just done unbiased screening, you would have missed many of these. One protein is particularly interesting, and I'll show you sort of going now from a discovery stage to a validation and then also clinically meaningful scenario for this particular protein. We talk about the beta-7 subunit of the proteasome, which turns out to be in many colon cancers highly overexpressed. and actually quite nicely shown here in this immunohistochemical experiment. As you know, the proteasome breaks down proteins. It's involved in protein degradation. I'll show you in a moment. It exists actually in 2 isoforms, and that's very important for that part of the story. But it's very highly expressed in nuclei, so there's a lot of protein turnover happening. There's some cytoplasmic, of course, proteasomes, but there's a high concentration of these in nuclei. And you can see that in these carcinomas, there seems to be an overexpression. So what does this mean functionally, perhaps? So I told you that the proteasome exists both as a conventional proteasome, which is basically just breaking over up proteins. You need to get rid of proteins in cells, right? Otherwise it would explode. So you make new proteins, old ones have to be put in a garbage. And that's really your garbage machine. And it's a phenomenal catalytic machine, really, a molecular machine that does that. It exists, however, also as the immunoproteasome under inflammatory and pro-inflammatory conditions, which in cancers, as you know, colon cancer, very inflammatory condition, often many other tumors as well. And what's interesting is that actually 3 subunits, the catalytic subunits of the proteasome change, and the beta-7 subunit is a critical one of the 3. And it becomes then, it changes the substrate specificity and produces peptides that are now presented on MHC class I molecules. So this is obviously done by the immune system to present peptides that are now antigens and hopefully perhaps neoantigens of the disease. So what's interesting is by stoichiometrically overexpressing the normal subunits, you can actually drive the system, even under inflammatory conditions, to avoid making the immunoproteasome. So even under inflammatory conditions, you're not able to present as many MHC class I presented peptides as you would otherwise do. So why would a tumor want to do that, you would ask, right? Well, obviously, if you can turn down immune recognition in the tumor, that's an advantage for the tumor. Now let's look at what this means in terms of survival. So we then looked at a pretty large independent validation cohort and just looked at overall survival in this particular case. And it turned out actually that the subclass, which is very interesting, of patients, and I'm just showing you females, but the ones who actually had received chemotherapy, in that cohort, the amount of PSP7 that was expressed, so if you had overexpressors, they did worse, as shown in the lower survival plot, which is really what the hypothesis was. So those would be tumors that are able to escape immune recognition better than tumors that are not able to make that switch. So since we made this observation, others have seen similar things in melanomas and other diseases where the same hypothesis holds that the switch between the immunoproteasome and the conventional proteasome is an escape mechanism. And I think this is a nice story because it highlights you're going from a somewhat unbiased but fractionated proteomic enrichment strategy to a validation to actually something that is meaningful in terms of outcomes, but also generates a hypothesis of an immune escape mechanism in colon cancer, which is obviously very important now for thinking about immuno-oncology interventions in tumors like that, where the tumor has an intrinsic ability to shut down the immunoproteasome. Let me switch gears a little bit. I want to talk about glycosylation events, so putting sugars on proteins, very important. Most of the proteins in our body are glycosylated for making them soluble, stable, and signal appropriately. Yet we have a hard time actually studying this properly because there are very few technologies that can study glycosylation properly on proteins. So functionally very relevant, but hard to study. So we used a strategy with lectins to actually pull down specific glycosylation events on proteins. So lectins are very specific. They bind to carbohydrates that are expressed on the surface of or attached covalently on the surface of proteins and can pull them down, and they actually have very specific specificities. And again, you can do that, and this particular study was done in lung cancer, and you can do this with different things like CON-A, for example, WGA, AIL. These are different types of lectins that pull down different kinds of glycosylated proteins. Again, long story short, a particular protein that came out as a biomarker is actually Phe2Na. This shows you that Phe2Na is actually in vivo glycosylated using a glycosylase. You can see there are 2 different bands. If I add the glycosylase, the protein loses molecular weight, cutting off the asparagine-linked glycosylation chains. We then used actually a Glycoarray analysis. That's an array on a solid phase that can again capture glycosylated proteins. You can look at different patterns of these proteins. And it turns out that actually high mannose content in Phe2Na was something we only found in cancer, but not in normal lung tissue. And the hypothesis there is actually, again, I don't have time to get into that, is that Phe2Na is an interesting antagonist of TGF-beta, So you can modulate TGF-beta signaling by that. And the hypothesis has been that differential glycosylation forms of Phe2Na have different binding affinities to TGF-beta. So by doing that, you have a modulation of signaling by a post-translational event that you would never capture by looking at nucleic acids alone. So I think a very nice story. And why is that important? I'll show you actually another example of glycosylation that came just out of Vanderbilt from our biochemistry colleagues there. And that's the story of PDL1. So PDL1 is also highly glycosylated, and in particular, actually asparagine 192 is glycosylated, which is shown here by a mass spectrometric assay that looks at the glycosylation event at this particular peptide and this location. So why is that important? And there are many people in the audience who are, and all of us measure PDL1 expression now by immunohistochemistry. And it turns out that actually this particular glycosylation event at that asparagine abrogates some of the antibodies that are being used. In this case, it's the E1L3N antibody. So what the lower panel shows is that this particular melanoma patient actually had essentially not detectable PDL1 with the antibody assay, but was actually the entire cohort, the patient with the highest PDL1 expression and total amount of protein present in the tumor. But all of that was highly glycosylated and therefore not recognizable by that particular antibody. I think this is a nice story just as an example of how PTM's post-translation modifications and their ability to detect them directly have a great advantage and really will help us understand what some of these glycosylation events mean. They also may explain why you have very different findings with other types of technologies that are antibody dependent. So now I want to switch gear again a little bit and tell you something about applying proteomics and mass spectrometry to discover antibodies and antigens in tumors. So this idea of neoantigens, again, something that's very hard in immuno-oncology, but again, our ability to really do this directly is very limited. And this comes actually from, and sometimes really helpful. So I'm board certified in laboratory medicine also. And a few years back, one of the faculty members got sick there, and I had to sign out ANA, antinuclear antibody assays for a couple of weeks. And it was interesting because that gave me the idea actually for all of this research. So when you do antinuclear antibody work, what you do is you take either cell lines or some test tissue and put the patient's serum on top. And then you detect the bound antibodies with a fluorescently labeled antibody, and you describe the patterns. So for example, this is homogeneous pattern, or speckled pattern, or nucleolar pattern. And these correlate with autoantigens present in this target cell line against which the patient has made antibodies. And this is widely used, and it's been around for decades. The problem is this technology is also not that smart because you really don't know what the antigens are. What is it doing? But it sort of gave me the idea to say, well, maybe we can use a similar but perhaps a little more sophisticated technology to start looking at antigens that are expressed in tumors versus normal tissue, and actually use the patient's own humoral immunity to help us discover those proteins. And again, it's nice because really sort of from an assay that's been around, that's not been improved, unfortunately. This assay, as I said, it's run millions of times around the world, but nobody's actually started to improve on it. But it can also obviously be applied to another disease besides autoimmunity. And so this is one example of how this works. So this is a patient, again, with colon cancer where normal colonic mucosa on the left column and invasive cancer in the right column is used. And then what's really the essence of the figure is the bottom two, E and F. On the left-hand side, you overlay this gel that contains the patient's proteins with the patient's own serum, and no reactivity happens. So that's good news. The patient doesn't really have autoimmune disease against the patient's own colon, which is good. But on the right-hand side, that's the patient's invasive adenocarcinoma of the colon, and you see lots of reactivities. The top is actually interesting, what I call control. That's using not the patient's own serum, but an unrelated patient actually with lupus with very high titers of autoantibodies. And you can see the patterns on the left and right, C and D, are similar. That means there are proteins, obviously, that are antinuclear proteins and so forth that are present in both of those samples. But it's really the comparison of E and F that's where the money is. And so, again, long story short, a number of potential biomarkers came out. One is MASPIN. I can't talk too much about, again, the function. We can talk about it later, perhaps, in the Q&A. But a number of proteins that are highly immunogenic, and probably because they're either overexpressed or abnormally displayed or in an abnormal context. Another protein, OLFM4, which is again very interesting because as I show you, it has prognostic implications. This is testing actually this particular immunogenic protein, both in the primary as well as in liver metastases, whether or not it retains its antigenic properties, and it does, and showing actually the expression of that protein in tumors. And actually normal colonic mucosa, it is expressed the base of crypts somewhere in the proliferative niche. That's its normal physiologic role. But you can see in many tumors, obviously it becomes highly dysregulated and perhaps also not properly presented. And I think that's one of the other reasons why it becomes immunogenic as a marker in cancer, not in normal situations. When you then again go from this discovery stage, you discover a potential biomarker, to a validation and independent cohort and ask, does it have any implications in terms of outcomes? Does it have any implications in terms of clinical features? You can actually see that in this case, I'm showing progression-free survival, that differential expression of this protein may have a predictive power to, again, predict outcomes, which, don't forget, this was not discovered originally to have that property. So you really need to check for these properties independently and figure out and test biomarkers in a very good independent experiment to be confident that they have these other properties beyond purely their biological function. So now I want to talk a few minutes about moving away from enrichment and targeted strategies such as heparin enrichment, glycosylation enrichment, or capture with antibodies, to actually something that's much more broad, something we call unbiased proteome profiling, where really I'm not enriching anymore. And this is becoming more and more feasible as instrumentation becomes more sensitive, and our ability to do online separation in very long liquid chromatography columns has really matured to perfection as compared to perhaps 5 years ago. So the way this works, and some people call this shotgun sequencing or shotgun proteomics, in analogy a little bit to what we would do in whole genome sequencing, is that you take an entire piece of tissue. You may purify, obviously, a component that you're more interested in. Then trypsinize it, basically fragment it, proteolytically cleave it, and then run it. And that's really the key here. High-resolution chromatography is where really the money sits. And then you use instrumentation to detect, in this case not hundreds, but tens of thousands, hundreds of thousands of peptides that come off in real time over the course of about 4 hours from the machine. Try to characterize as many of them as you can, and quantify as many as you can. What's coming off in analogy to any other chromatography is called an ion chromatogram. So these are different samples, and time on the columns are from 0 minutes to about 4 hours. And under each of these peaks are literally thousands of peptides. So this is just showing you the total intensity. It's like a chromatogram that would do any other column. But underneath is a lot more, and our ability to detect it allows us then to ask the question, what's under each of these peaks? And so you can detect now from relatively little inputs, from about 2 micrograms of protein extract from human tissue, you routinely can now detect at least 2, 5, maybe even 8 to 10,000 proteins, depending on how you set up your experiment. And so then what you get, you not only get a list of the proteins that are present, and again, this is unbiased. You're really not saying I'm limiting myself to proteins of a certain a feature or a certain biochemical modification. Again, this can be done from relatively few cells. In this particular case, maybe 200,000 individual cells. And you get pretty good quantitative information about it. So you can say relatively, is one 100-fold higher than the other, or up to perhaps 10,000, 100,000-fold. As I said, about 10 to the 5 is your dynamic range here. And this is sort of the analogy as if you were trying to do an immunohistochemical, any antibody detection assay, with trying to come up, to measure targets that are in the thousands. The problem is you really don't have that many, and you really would have a hard time doing this multiplexed. The added benefit that I don't have much time of talking about here is that you can actually also detect mutations. So if you know what you're looking for, you can find your KRAS mutation, your P53 mutation. It works for SNVs much better than for large deletions, or of course proteins are truncated and not made. you can start detecting them and actually even ask the question, is the mutation that I'm seeing at genome level even making it into a protein or not? Which remains one of the big unanswered questions in biology. So yeah, this is just to show you how that would do. So you can pull out data from these experiments. You can look at macroproteins, for example, MAP kinase, so things that you have antibodies for routinely and things that you don't. And you can get these lists, basically what we call deep proteome profiling. You get a list, a personalized profile of that patient's tumor. What's high, what's low. Actually, the first protein that shows up is IDH1, actually, interestingly. So there are proteins of very high biological significance in this list, not just cytoskeletal proteins. So I want to end the talk with thinking about how we can move some of these technologies and concepts. And again, I'm showing us different technologies. I think it's not one technology that answers everything. It's rather, we have an interesting biological question. In this particular case, I think we as pathologists are looking at how is the next generation of clinical trials and drug development improving and changing, and what are the technologies that we can bring to bear that will help this and improve and really change the game for clinical trials. And so if we were to bring in proteome technology into clinical trials, what would we like to do? Well, we would like to have high sensitivity tests, both for the clinical trial as well as obviously for preclinical development. We will need samples that are optimally collected and ideally serially. And that's already when you look at ongoing clinical trials where we have a real problem. There are very few trials right now ongoing where we actually have good serial sampling, tissue and bloods, of patients before, on, and off treatment. But if we brought technology like this in, this will really allow us to look at that, and I think it needs to be a joint endeavor between technology development, diagnostics, and advance, as well as thinking about clinical trial protocols that are optimized to these technologies. Another example that I just show you, you can do antigen discovery, as I showed with the antibody antigenome discovery, but you really need good paired serum and tissue samples to do this, and you need well-timed sampling for that. Another thing that's really exciting for plugging such technology into an ongoing clinical trial is that we can start calling mutations and other aberrations that we know exists based on DNA or RNA information, but about which we have no idea whether they actually make it in the protein domain. And that, of course, in itself is really very critical for understanding whether a drug works or not. So if you have a mutation, but the protein's not made because it's unstable, even the drug that's supposed to bind to that mutated phenotype will not work, obviously. So knowing that is critical, and technology to do this and to bring it into clinical trials, I think, will be a game changer. Another area is to really think about dynamic plasticity of the proteome. One of the really attractive things of looking at proteins is that they very rapidly change. When you start drinking alcohol, your liver actually starts inducing those enzymes that break down the alcohol very, very quickly, within minutes. And then some people better than others, and so forth. But what this also means is that if you extrapolate that to human disease, we start exposing a certain tumor to a drug, different tumors will respond differently. That's what I mean by dynamic changes. These are, again, things that are not encoded at nucleic acid level, but need to be studied at a functional protein level. And again, doing this within the context of a clinical trial, I think, is going to be key. So I'll just show you something. So we've started to plug in some of those thoughts, just ideas from the previous slides, into an ongoing multi-center pediatric trial. So it started here already. Stanford Children's Hospital will come on next, and then there were a couple other children's hospitals. It will be the hub receiving those samples. A lot of things will be happening with these samples, including looking at protein aberrations. And so the idea is, develop this, do this in a timely manner, do this with samples that come from actual trials. Is all much more challenging than doing them with idealized samples that you can control in the lab. So this is really just getting our hands dirty, if you will, but it's generating data. So this is an idea. This is basically one of those lists that I showed you of deep profiling of a couple of those patients in the trial. One particular patient with fibrolamellar HCC, for example. And then just looking and saying, well, if you took total protein profiles, just abundances, and didn't even know what the diagnosis was. So I put those in, and this is just an example. I have no idea what I was looking at, which of course we do, but just to look how different are these things. It's actually quite nice that these diseases group together. And I think that's a good example. And we've learned a lot really in terms of how much sample do we need, how do we need to process it, how do we start thinking about integrating this information with, in parallel, ongoing sequencing that's happening on those samples. So that's yet a question to be answered, but one that I'm very interested in. So in closing, we talked about really strategies to discover protein-based abnormalities in human disease, strategies such as enrichment to look at things that are particularly rare or a little abundant but of interest. How do we get at them? I was talking about approaches to look at protein glycosylation using lectins and mass spectrometry, and how this is relevant to understand sometimes discordance and understand performance criteria of antibodies that don't recognize epitopes that have differential glycosylation. And in many instances, of course, differential glycosylation has biological properties because proteins aggregate and associate differently, are differently active. So these are things that you can only study when you do it that way. I talked a little bit about applications in I/O, on how we can try to identify neoantigens, which is really the hottest kid on the block right now, is trying to understand what neoantigens characterize a tumor and how are they activating or not activating the patient's immune system. Unbiased profiling is really just characterizing as much as you can without restricting yourself to a particular molecular entity up front. And then a few thoughts about how a next generation of clinical trials can be built where we can plug in technology and questions like that in a better manner. So I want to close with sort of this idea that I'm very interested in, and obviously lots of people in the field want to see this move forward, is to integrate information that comes from nucleic acid-based analyses and informing studies at protein level in a better manner. And the NCI is trying to do that as well, and that's known as the Clinical Proteomic Tumor Analysis Consortium that has started. I'm happy to say we're part of this now, once we get it through the lawyers at MSK. But no, we're part, and this is a big initiative that the NCI has really started, exactly understanding, we need to go a step beyond. We need to understand what's the functional implication of aberrations at the genome level, what do they do at protein level, what are the technologies we need to bring to bear, and really doing a large-scale effort. This is, if you will, a next step. This is a proteogenomic evolution of the TCGA, and I'm very happy that that's ongoing. So I'm at the end of my talk, and thanks very much for your attention, and I'd gladly take questions if there are any.